My name is Gaurvi Awashti. I am a rising sophomore at Cornell University and a bioinformatics trainee at the Mayan Lab at the Icahn School of Medicine at Mount Sinai. Today, I will be presenting on identifying immunotherapeutic targets for non-brain solid tumors with Target Ranger. To start off with some background, Boss et al. Developed a RNA sequence-based process that identified a cell surface protein that is differentially expressed at high levels in neuroblastomas compared to normal tissues from GTEx. In this way, GPC2 was identified as an oncoprotein and a candidate for immunotherapy in high-risk neuroblastomas. Inspired by this, the Mayan Lab developed Target Ranger, a web server that enables users to input RNA-seq data from a tumor. Then using data from proteomics and transcriptomic atlases and filtering for membrane or secreted genes, Target Ranger outputs targets that are highly expressed in the inputted RNA-seq data from the tumor and lowly expressed in the normal background. My project aims to further improve Target Ranger by identifying genes that are highly expressed in normal brain tissues and also highly expressed in non-brain solid tumors. This is an overview for my project. We aim to identify potential targets for immunotherapy by first inputting RNA-seq data from the TCGA database. This data was compared with the background data of normal gene expression tissues from GTEx. This is the process that is currently implemented by Target Ranger. The new analysis path also considers gene expression data from GTEx, except it does not contain any brain-related tissues. The outputs of these two paths were further analyzed to identify additional targets that emerged after removing brain-related tissues from the background. In addition, it is important to discuss that the reason that brain tissues are removed is because of the blood-brain barrier. The blood-brain barrier is a protective layer comprising three main layers, shown in detail to the right. Overall, it is responsible for the vascularization of the central nervous system as it supplies blood and necessary nutrients to the brain, while also preventing macromolecules and neurotoxins from entering and reaching the brain. As mentioned before, the RNA-seq input data for this process is from the Cancer Genome Atlas, or TCGA, which is a cancer genomics program that molecularly characterizes 33 cancer types. In addition, the Genotype Tissue Expression, or GTEx program, is a member of the Common Fund Data Ecosystem, or CFDE, which provides open access to gene expression data from 54 non-disease tissues across 948 donors. In this project, the GTEx data serves as the background data for gene expression in normal tissues. In order to remove the brain-related tissues from the GTEx background, brain-related terms such as brain, cerebral, hippocampus, amygdala, and hypothalamus were used to identify and filter out these tissues. The first step in analyzing the TCGA data was to apply the Leiden community detection algorithm in order to identify clusters amongst each of the cancer types. For each cancer type, this process yielded about 2 to 15 clusters with 4 to 390 samples within each of the clusters. The graph below shows a UMAP of the 5 clusters for adrenocortical carcinoma, or ACC, that were identified using this process. This part of the analysis was done by Giacomo Marino, a bioinformatics software engineer in the Mayan Lab. Next, the metadata acquired from the TCGA database was used to create Kaplan-Meier, or KM curves, that estimate the prognosis for each cluster. In this plot, cluster 2 has the poorest prognosis, so we decided to focus on finding novel targets for cluster 2. We looked for targets that moved up in rank by at least 50 spots. These targets were then compared with the other adrenocortical carcinoma clusters to determine which ones were unique to this specific cluster. In this case, it was found that ATP2B3 was used when it was unique to cluster 2 of adrenocortical carcinoma. In this process, this was found, and this was also repeated for all the other clusters in all the other cancer types. This analysis yielded the following 20 targets as novel potential immunotherapeutic targets for each of the associated cancer types. These 20 targets were further analyzed to determine how well they were already known. This was done by searching publications on PubMed. This figure shows the PMIDs for different search parameters, demonstrating how well the 20 targets have been researched already in general, in the context of cancer, and in the context of the specific cancer type. Looking closely at this plot, although many of the genes have been researched, the gene and its associated cancer have not. So there's a potential new connection between the discovered target and the cancer types. For example, SLC7811 was found to be associated with bladder urothelial carcinoma, or BLCA, through this project, but in prior work, it has been associated with diabetes, ferroptosis, and other cancers such as lung cancer. Therefore, although SLC7811 is well studied, its association with bladder cancer is not. Towards the center of the graph, we see that ATP2B3 has been studied in the context of adrenocortical carcinoma. Looking to the right of the graph, we see that there are a few understudied novel targets as well. In other words, genes that should be further investigated as immunotherapeutic targets for cancer. From there, we focused on the following 5 of the understudied novel targets. Further analysis of these genes included examining their expression across normal tissue and the associated tumor. The figure on the left shows the expression levels of GPR158 across normal tissues and in bladder urothelial carcinoma, and the figure on the right shows the levels of PODXL2 across normal tissues and in kidney renal papillary cell carcinoma. In these figures, you can see that the expression of the target in the associated tumor and in normal brain tissue is relatively high, while it's lower among all other normal tissues. Similarly, in these plots, the expression levels of the other 3 novel targets are shown. Here too, these genes are shown to be highly expressed in the associated tumor and in normal brain tissues, while lowly expressed across non-brain normal tissues. We repeated this analysis on the CPTAC data, still using GTEx as the background data. This analysis resulted in the following 3 potential target genes. These plots show the expression levels of TAS2R4, GJB1, and GPR75. As before, the genes are highly expressed in the tumor and in the brain tissues and lowly expressed across non-brain normal tissues. Overall, in this project, we developed a method that extends target ranger to be able to identify novel targets by implementing a brain filter. In the future, the brain filter will be added to target ranger, and this method can be applied to other cohorts as well. For example, to the data from the Kids First Hope project. This process yields novel targets that can potentially be targeted for immunotherapy, for example, by developing ADCs or CAR T-cell therapies. However, it is also important to note that since these genes are highly expressed in the brain, such therapies need to be developed in a way that the therapeutic agent will not be able to cross the blood-brain barrier. Finally, I would like to thank all of the members of the lab, especially Dr. Mayan and Ms. Sherry Jenkins for giving me this opportunity. I would also like to thank Giacomo, Daniel, and Eden for their contributions and support throughout this entire project. Thank you so much for listening, and please leave any questions in the Q&A section. The next speaker is Mason Zhang, who will be discussing an age prediction model he has developed over the summer.